much. And I wanted to thank the organizers, and I wanted to thank the staff at Rice and Sarah for organizing the organizers. And and I want to thank you all for sticking it to the bitter end. People are going to think these are not very exciting people across town, that they were coming back for this after lunch. So critical proteins involved in DNA repair that traffic on microtubules, and it'll go beyond that. Yesterday, several people were saying, I don't know how this gets to the nucleus. I'll show you how it happens. So it's been almost 60 years since we gave vincristine, which is a microtubule targeting drug, to children and adults with cancer. It was 1961. And Susan was talking about not knowing how something works after 10 years. Actually, it took us about 50 years to figure out how microtubule targeting agents work, and most people still don't understand it. So microtubules, I'm surprised there isn't more physicists interested in this molecule. Yeah, I mean, it's a really cool molecule, right? So there's alpha and beta tubulin, and alpha and beta tubulin are one next to the other, and they form these long protofilaments, and then 13 of them, what in the US we call a baker's dozen, organize into something that has a lumen down the middle, which is a microtubule, and that's what keeps cell structure, and that's what's important in cell division. So again, it goes to this, and then it becomes a microtubule, and there's this powerful association of these protofilaments, and this can be 1 million of them long, which is amazing. And they can form, and then they can disaggregate in a matter of seconds. Now, the way that we usually think about microtubules in cancer is, okay, they're important in mitosis, and if you have a drug that targets mitosis and disrupts mitosis, well, cancer cells divide rapidly, we're going to work. As a cancer drug, that's not true. So in cell culture, there's no doubt, you take cells and you treat them with Taxol. 24 hours later, all the cells have arrested in G2M. They get to the point, this is, G2M, they're really arrested in mitosis. They have a G2M, a 4N content of DNA. In animals, where tumors also grow very rapidly because you want that answer tomorrow, so next week you can get it into a patient, and next year have a lot of revenue if you're a drug company. So in animal models, this is the one mitotic figure that you find after we add 2-methoxyestradiol. Every cell is arrested in mitosis. In this case, it was 72 hours after doing it. But in rapidly dividing cells, that's what happens. And if you go back and look at that original paper describing the 1961 experience, they were talking about these cells induce metaphase arrest, and that sort of put it in context. But really, what about in a tumor that is inside a human? And so I first thought about this in 2001. Had a patient come to the National Cancer Institute, and she had cervical cancer, and it took us 4 months to get her enrolled on the clinical trial. And we had scans at the beginning, in the middle, and at 8 months before she enrolled. And over that time period, the tumor hardly grew. Had it been dividing basically once a month, a 1 centimeter tumor would have been about 16 centimeters cubed. As it had been 5 days, it would have been 4,096 centimeters cubed. And if it was a 1-day division, as it is in cell culture and in animal models, it would have been 1.6 million centimeters cubed. I would say the patient wouldn't have fit in the cancer center. The tumor would have been too big. And yet, when we finally did start treating her in August, and she came back 6 weeks later, the tumors that we were observing had this much of a decrease. And then you say, wait a minute, this hardly doubled over this period of time, and now all of it has gone away. Did we just magically induce mitosis and arrest mitosis? No, that's not what was really happening, because patients on this and other studies, and this is 4 breast cancer patients, we gave them this drug which is called ixabepilone, which is like Taxol. It arrests cells in mitosis in cell culture. We gave it to them on 5 successive days, and right after the 5th dose, so it was 2 hours after the 5th dose, 24 hours after the 4th, 48 after, we didn't know what was the right time to do it, so why don't we do it on the 5th day? And we cover from 4 to 96 hours. You can see how many mitoses aren't there? And in fact, if you look in the literature, there isn't a single example, with one exception, when Taxol was being developed, there's a leukemia where they show one picture of one cell arrested in mitosis. In humans, we never see mitotic arrest. So, and why? Because although if you have a loved one with cancer, they die all too soon, most tumors don't divide very rapidly. And this is data from the 1990s and 1980s when we didn't have cDNA arrays and all of our fancy things, and people would just measure things on x-rays and so forth. And these are the doubling times, and they're very large, very, very large. Obviously, if you're dividing 203 days, if you trust me that once you have metastatic disease, 4 divisions leads to death, you're not too far away from dying. But it doesn't grow or double every day as they do in cell culture. Then there was this other interesting observation, and that is that in 2004, the Food and Drug Administration in the United States approved Taxotere, another microtubule targeting agent, for the treatment of prostate cancer. The average patient with prostate cancer who's more than 70 years old, you usually think you're not going to die of prostate cancer, you're going to die of something else. We'll soon have to raise that to 80 because we're living longer. And why? Because it grows so slowly. And then you say to yourself, so really? We approved the drug. It was the first time a chemotherapy drug had been approved in prostate cancer. Prior to this, only hormonal drugs had been approved. And the first chemotherapy drug that we approved for the slowest growing tumor is a mitotic arrest drug? No, there must be something else to this. So, and then finally, there was a period some years ago when there was a lot of enthusiasm for what we'll call mitotic kinases, proteins that are in the nucleus that are involved in mitosis. And people said, we're going to interrupt the function of these proteins and we're going to cause mitotic arrest. And they said, that'll be fantastic. Because what happens with Taxol, with vincristine, with Taxotere, with all those drugs, is that you get what we call peripheral neuropathy in your feet first, and in your arms and your hands second. And that's because, you know, at the tip of my toe, the cell body is back here, my spinal cord someplace. And it has this huge long axon through which things have got to go and feed my nerve down at my toe, and that traffics on microtubules. And if you interrupt microtubule trafficking, you all of a sudden start to see that you get toxicity. And in fact, you get toxicity in the one tissue in the body that hasn't divided since you were born. That nerve going to my left toe is the same one that was there the day I came out of the womb. So here we have a cell that doesn't divide and we get toxicity. And what did it have to do? It had to do with trafficking. But in any case, drug companies usually don't pay attention to things. So these are these mitotic kinases, Aurora A, Aurora B, and polo-like kinase and kinetic spindle protein. This became a $10 billion experiment. What we could have done with that 10 billion, right? Actually, after I stopped counting this, because I got tired of it, about 10 more drugs were developed. And it didn't change. There were 15 aurora kinase inhibitors, 5 PLK inhibitors, 5 KSP inhibitors. In all, there were 25, and I tell you, 10 more subsequent to that. This didn't change. The response rate in solid tumors, Susan was showing you 30, and she was saying, well, it could be better, was 1.1%. And the response rate in hematologic malignancies was 7.5, and these were usually patients with rapidly growing leukemias that, in fact, are dividing frequently enough that you could have some impact. So then you might say, well, maybe the drugs weren't good. Maybe that's why they didn't work. No, it turns out that the people that are good are not only the physicists, the chemists are really good. They've gotten to where they develop wonderful drugs. All of these drugs led to massive interference with bone marrow function because our bone marrow is constantly dividing. In fact, every day, your bone marrow and my bone marrow makes the equivalent of 5 2-centimeter tumors worth of white cells. So just think about that, okay? That's more than 15 mil Eppendorf, okay, of new white cells. And if they don't do that, in 10 days you don't have any. So you start these drugs, 10 days later, you don't have any white blood cells. So in other words, the drugs were doing what they were supposed to do. They were inhibiting the mitotic kinases and leading to arrest of cells that were rapidly growing. But that wasn't the average tumor. Okay, so if it's not mitotic arrest, what is the mechanism? There's only one other thing you can look at, and that is the microtubule in interface. So what it turns out is that in prostate cancer, the one thing prostate cancer likes more than anything in the world is androgens, testosterone. They just thrive on it. It turns out that the androgen receptor gets to the nucleus where it turns on all those genes that make prostate cancer bad by trafficking on microtubules. So in fact, what we were doing when we were giving patients with prostate cancer Taxol, we were actually interdicting the trafficking of that all-essential androgen receptor. And that's how the drug works. And then there was this observation. These are drug combinations that even in the immunotherapy era are used in every patient. And in fact, immunotherapy has been good in some things, a disappointment in the majority. And where it's been good, it's being combined with these drugs. But when you went back and you looked at what were we treating all of these cancers, and these are the recommendations for these cancers, invariably you saw a DNA damaging agent and a microtubule agent. The blue is the DNA damaging agent, the red is the microtubule agent. So then you say to yourself, so was this a combination of a DNA damaging agent and a microtubule agent fortuitous, or was it based on some mechanism? So what we said, or what we thought was, well, maybe these DNA repair proteins that have got to quickly repair the damage are being interdicted and can't get to the microtubules. So we actually knew from data we had published in 2000 that this was true for P53. Here's P53. If you treat with adriamycin, the amount of P53 in red goes up dramatically. And where does it go? It goes to the nucleus. And there you see P53 in the nucleus. This yellow is because the green is tubulin. All of the P53 that is in the cytoplasm isn't floating around, it's sitting on the microtubules. It's trafficking to the nucleus. And you can see here again, treat with adriamycin. There you see it, and in the cytoplasm, it's all latched on the microtubules. And if you treat it with a microtubule targeting agent, in this case Taxol, and adriamycin, P53 goes up. Here's the control. Here's after treating with adriamycin. Rapid rise in P53. The cell knows I've been damaged. I've got to do something about this. I've got to turn on the guardian of the genome, P53's best name, and get it to the nucleus. But if you treat with a microtubule targeting agent, in this case Taxol, it polymerizes the microtubules. It traps the P53 in these polymerized microtubules, never makes it to the nucleus. And what you see is that you get a lot more death in these combinations. What you also see is that these P53 inducible genes, such as MDM2, aren't induced when you've pretreated with a microtubule targeting agent. So we said, okay, so maybe those combinations between a DNA damaging agent and a microtubule targeting agent, that's because they work well together. So I told the people in the lab, okay, find 6 to 8 DNA damaging repair proteins for which there are good antibodies that work well on Western and also on immunohistochemistry and immunofluorescence. And so when they got to 9, which was the first 9, because these are widely studied proteins, I said, okay, that's enough. So if we were right, all of these predictions, and I'll go over them one at a time in subsequent slides, should be true. So the first thing was, and here it's really hard to see, this actually is yellow, so is this, so is this. When I project it like this, it comes out looking green. In every case, DNA PK, P53, P63, you saw how this was totally yellow in the previous things. For some reason, it doesn't project well here. The proteins are associated with the microtubules. They're all trafficking on microtubules. If we then take denane, or dynein, which is a motor that carries things along microtubules, and we do an immunoprecipitation and we say, what was denane holding on to? you find that it's holding on to all of these things. All of these proteins are trafficking on microtubules on denane, whose function is to take things in the direction of the nucleus, and they're all can be found when you bring down denane with an antibody and say what was attached to it. If you look at the amount of protein in the cytoplasm, when you pre-treat with a microtubule targeting agent, the amount of protein goes up. And finally, what we did was we used one of these aurora kinase inhibitors. And what you see is that with the aurora kinase inhibitor, there's no change in the amount in the cytoplasm, but there is when you use a microtubule targeting agent. And then do you see, synergy? Yes. In this case, the DNA damaging agent is radiation, irradiation. And what you see here is we're reading out DNA damage by looking at staining with gamma H2AX, which recognizes double-strand breaks in DNA. And when you pre-treat with Taxol, you get much more, and you can see here with Taxol and without Taxol, the area under the curve, if you will, of gamma H2AX, you get much more DNA damage that is prolonged for a greater time. So that it satisfies all of this, and we think that that's why, by chance, no, by taking us there, we ended up with all of these combinations that are a DNA damaging agent and a microtubule targeting agent. So then what we said is, I'm going to skip these two because I thought Susan was going to have gone over this, and you didn't. So, but what we then did was, okay, dynein takes proteins in the direction of the nucleus. The kinesin family takes them away from the nucleus. So what we did was, we did a dynein IP, immunoprecipitation in cells, and we did a kinesin immunoprecipitation, and then did mass spec analysis to see what proteins did I bring down. So rather than what we were doing before, where we had antibodies and we went looking for it, this was somewhat agnostic. We said, what came down? In fact, about 2,500 proteins came down in robust quantities. The majority of them came down with kinesin and dynein. And although a DNA damaged protein might be going to the nucleus, when something moves in the cell, it's never one directional. You see that they go and they come back for a while, and then they go and they come back for a while. It looks erratic, but at the end, you end up where you want it to be. And so they transfer from one carrier, one motor protein, from dynein to kinesin and back. And so the majority of them would come down with both antibodies. And this basically shows that there were some that were exclusively kinesin, some that were exclusively or nearly so dynein, but the majority of them were being carried by both dynein and kinesin. And then you said, what kind of proteins are these? This is the relative abundance to the amount of immunoglobulin that we had in the immunoprecipitate, which is the immunoglobulin we put in to bring down the dynein or the kinesin. And you can see that there's proteins that are not too abundant, and many proteins that are very, very abundant. So it was the range of these proteins was quite broad. So then, at that point, the question was, is this real? After all, we did this, I handed it over to my collaborator, Udaya Guha, and then he comes back and he gives you this printout. He says, you know what you're doing, Udaya, So what I said is, okay, let me look at the top 100, start there, and see whether or not the proteins that we brought down have been identified in the literature somehow associated with dynein, kinesin, microtubules, something. You can't see it, you don't have to. The ones that are white, I couldn't find anything in the literature. Every other one, you found something in the literature. So 90% of them. There was already something in the literature that when you went to Google and went to PubMed and so forth, could find that these proteins were indeed associated. So I thought, we're doing pretty well. I think this has been validated. That's the rest of them right there. So if you go to Wikipedia, and actually every month in Cell, Cancer Cell, Science, Nature, invariably there's an article that talks about how microtubule targeting agents arrest cells in mitosis. So it hasn't been driven home yet. So, but it says in Wikipedia, of course, separating its chromosomes during the metaphase, the cell that undergoes apoptosis stops the cell from separating the chromosomes during mitosis. And that's how they work. No. They work because a ton of critical proteins are trafficking on microtubules, and if you disrupt that, those critical proteins that the cancer cell is depending on, their function, their getting to where they have to be, is massively disrupted. And it's not just DNA proteins. This is the remodeling of epithelial adherence junctions, okay? And these are proteins that you know, Herbie, in going from EMT transition, the catenins, cadherin, all of these traffic on microtubules. If you look at a tight junction or a desmosome, the microtubules go there. And you thought, okay, that's holding on to it. No, that's how it delivers these proteins to the surface so that they can load then onto the surface and how they work. And I added this because in those first 100 proteins, there were a bunch of DNA repair proteins, all of which were validated. And actually, if you look in the 2000, it's just full of DNA repair proteins that have been brought down in the immunoprecipitation. And additional nuclear proteins that maybe we don't consider as important in DNA repair, but are involved. And then because this morning, actually, FUS, that we were talking about this morning in ALS, that also traffics on microtubules. High molecular group binding proteins, they traffic on microtubules. And RAS proteins traffic on microtubules. That's how RAS gets to the membrane to get loaded onto the membrane. So these critical proteins are all trafficking on microtubules. And why are these drugs important? Because they've been important in the past, and they're going to be important in the future. What's happening is that people are taking microtubule targeting agents that were so toxic that when given to a patient, the patient couldn't tolerate it, and binding it to antibodies or smaller molecules, and using this to deliver it to the cell. And TDM1 stands for trastuzumab, which is Herceptin, which is a drug that targets HER2 in women with HER2 amplification breast cancer. And TDM1 carries in this DM1, which is a derivative of a microtubule targeting agent, maytansine, that is extremely toxic. But by doing this, it's not circulating free in the circulation. It only goes into the cells, it goes to the lysosome, it gets released, and it has activity. And actually, what led to approval by this in the FDA was that women whose tumor had grown through trastuzumab were then given TDM1, and 40% of them had shrinkage. And it wasn't because of the trastuzumab, because the tumor had been growing. It was because of the DM1 that they delivered. And so that's how these drugs work. It's not by mitosis. It's by inhibiting the traffic of every essential protein. All of you have some protein that's really cool. There's a really good chance that it's trafficking on microtubules. And so these are the people who have helped me. And with that, just remember what the mechanism of action is for microtubule targeting agents, and that's all you have to remember from today's talk. Thank you very much.
Okay, we have another microphone somewhere? Oh, you have it? Okay, go ahead, Preston.
Yeah, I have it. So it seems that you have actually, frozen the cell, according to your hypothesis. Nothing is trafficking. But in order for a cell to commit, to go through apoptosis, you need P53 to activate cytochrome C, and most likely the way cells die through the mitochondrion. Now, if you can transport P53 anywhere, how does it commit? How does it go through apoptosis?
So what you're doing is you're delaying the process, and that's really what's happening in patients. Eventually, the drug is metabolized, and those cells that were able to hold on can then at that point repair the protein, the damage, and bypass it. But what you're asking is, how do the ones that die? At that point, then there's way too much DNA damage. It's unrepairable, and the signals for apoptosis are being sent out, and the process. The what?
The signal is P53.
It's P53, right. P53 goes in when the microtubule targeting agent eventually wears off, and then it finds all of this and it says, oh my God, we got to apoptose. So.
Dave?
I was a little, maybe more than a little lost in your description. There are a lot of proteins that are trafficking on microtubule. And I want to ask the question this way. Are these proteins mobile on the microtubule? And that's number one. And then if they're mobile on microtubule, then I also don't know what dynein is doing in terms of how it moves, because these could serve as obstacles or roadblocks. And how are these coupled in this trafficking scheme that you
Right. So both dynein and kinesin family actually walk on microtubules. There's a lot, and if you go online and you do this, you find these cute little movies that have been made
all
up. and-
incorrect movies, but I've seen them. yeah.
Right, yeah. And then dynein, then between dynein and whatever the cargo is, there's several adapter proteins, some heat shock proteins and some other things that sort of bind that. And then basically, with P53, we know this. Dynein is loaded in the cytoplasm and then jumps on the microtubule and carries P53 in. And the reason we know it is because we made a mutant that prevents P53 from tetramerizing, and it has to tetramerize first before it can bind to dynein and be taken to the nucleus. And when you do that, it all remains in the cytoplasm, and dynein is in the cytoplasm.
But the proteins are not only transiently interacting with the microtubule.
Right, exactly. And then when it gets near the nucleus, it offloads it, and then there's nuclear import proteins that take you the rest of the way.
Thanks.
I guess I'm going to ask a question, then I'll get to. If you give these microtubule drugs, do you get incidental tumors the rest of the body? Does it go way up? I mean, when you do, in other words, how tumorigenic or whatever are microtubule agents if in all the cells of the body you're basically for some amount of time blocking DNA repair? That must be a huge source of possible other arising cancers. Is that seen?
Yeah, so there's actually two sets of experiments on that. In the early 1960s, at the NCI, they were treating patients with leukemia with doxorubicin or adriamycin and with vincristine. And they took out the bone marrow, the normal marrow, on these patients, and they did cytogenetics, just heme sustaining. They're amazing pictures. The amount of DNA damage that is there is incredible. And you look at that and you say, why doesn't everybody who gets chemotherapy get leukemia? And which is what you're asking. And I think it's just that normal cells have a low threshold for apoptosis. And there is so much damage that they just apoptose. So almost you're self-protected. For the damage to be enough to get leukemia, it turns out that that's usually so lethal that the cell dies, doesn't survive, and then never gets a chance. The what?
You'll be sick as hell, right?
Yeah, I mean, so, you give these drugs to somebody and 10 days later they have no white count, And so, yes, it does make you sick, but, yeah.
Yeah.
And then, I'm sorry, and then the other thing, we actually did this in monkeys around 2000. We gave these drugs to monkeys, and the microtubule agents, and then did bone marrows on the monkeys. Had a pediatric fellow working with us, so I thought, monkeys, kids, these are about the size that he's used to treating. He can do bone marrow on these monkeys. He did. And adriamycin, huge amount of DNA damage. Etoposide, huge amount of DNA damage. Vincristine, a lot of DNA damage, not quite as much.
Yeah, that was more or less my question was on the side effects, because in one of your slides you showed at the bottom, yes, it did arrest the cancer cells, but it looked like a real impact on the intestinal epithelial cells.
Right.
And one other one. So why those particular?
So the toxicity is usually bone marrow, because as I told you, our bone marrow is on steroids. It's really pouring it out every day. The GI tract divides very rapidly. And in fact, mice are incredibly sensitive to the zinc alkaloids, such as vincristine, because it's dividing so rapidly. It's actually turning over every day, basically. And the third thing is those neurons that I was telling you about that need their nourishment that originates in the cell body to traffic through the microtubules, but you've disrupted that. And eventually, you do get axonal loss in somebody who gets these agents. But the first thing that you do, they actually get numbness and tingling, because these cells are dying and they're just firing off, erratically. And then all of a sudden, the numbness and tingling goes away because the cell eventually dies. And we've got so much redundancy in our fingers and in our toes that even if we lose 10, 20, 30% of them, eventually you don't realize it. So,
Okay, well, thank you again.
okay.
So